Imagine you decide to take one of those genetic tests.
Oh, right, like the mail-in ones.
Yeah, exactly. You know the drill. You spit in the tube, you seal it up, drop it in the mail, and then, like, a few weeks later, you open the results on your phone.
And you're expecting this very clear-cut answer about your health.
Right. But instead of a clear answer, the report just says you have a variant of uncertain significance, a VUS.
Which is just the worst feeling. It's so vague.
It really is. So you take this report to your doctor, and they essentially just, well, they shrug. They tell you they don't know if this variant is going to make you sick or if it's just a completely harmless, quirk in your DNA.
Yeah, it's a uniquely frustrating gray area for patients.
And the reason we get stuck in that gray area, and this is what we're really getting into today, is that traditionally we've looked at genetic mutations as this, like, simple binary switch.
Exactly. Good or bad, on or off.
Right. A mutation is either benign, meaning it's harmless, or it's pathogenic, meaning something is broken. But what if a mutation isn't just a switch? What if instead that single mutation is altering the literal physical architecture of your cells in these completely unique, multidimensional ways?
Ways that up until very recently we couldn't even see.
Exactly. So today we are doing a deep dive into this groundbreaking 2026 paper from the journal Cell, because this paper, it essentially shatters that old binary way of thinking.
It really does. It's a total paradigm shift. The researchers behind this work developed this massive new mapping technique. It's called VIS-seq.
Which stands for variant in situ sequencing, right?
Yep, that's it. And what makes VIS-seq so revolutionary is that it proves mutations have a tangible spatial reality. It moves us entirely past that simplistic benign versus pathogenic model.
I mean, the actual title of the paper is quite the mouthful, I won't lie. It's something like image-based pooled phenotyping reveals multidimensional disease-specific variant effects.
A very classic, dense science title.
Right, but essentially what they figured out how to do is take high-definition 3D mugshots of millions of mutating cells all at the exact same time.
Millions. It's an incredible scale.
It makes me think about how we usually test genetic variants with that process called deep mutational scanning.
Yeah, the standard approach.
It feels a bit like the check engine light in your car.
Yeah.
Like it turns on and tells you something is wrong. Maybe a cell stops growing or some protein level just drops.
But it doesn't give you the mechanics of the actual failure.
Exactly. It just says engine bad. But VIS-seq sounds more like, I don't know, plugging your car into the mechanic's full diagnostic computer while simultaneously getting this, like, glowing 3D hologram of the engine while it's actively running.
That analogy actually captures the technological leap perfectly. But, we have to push it even further to really understand the biology here.
Okay, push it. How so?
Well, current methods frequently fail to parse these complex gene-disease relationships because they only measure one-dimensional traits.
Like just checking if the cell is alive or dead.
Exactly. But a single gene can have thousands of different possible mutations. If you only look at whether a cell lives or dies, you completely miss the nuance of how it's dying or how it's misbehaving structurally.
Right, the actual physical mechanism.
Right. By physically looking at the cells and extracting these dense computational features from their structure, VIS-seq allows us to connect the dots. We can connect a microscopic genetic change directly to a macroscopic physical disease.
We can literally watch how a single typo in the DNA cascades from a molecule to a structure to a disease state.
Exactly.
Okay, so I want to get into the weeds on how we actually pull that off. Because taking a 3D mugshot of millions of cells sounds like a complete logistical nightmare.
No, it's a massive challenge.
How do you physically engineer a test like that?
It's just an incredibly elegant piece of engineering. So to prove the concept, the researchers selected about 3,000 different variants.
So that's 3,000 distinct mutations.
Yes, across two highly medically relevant genes, LMNA and PTEN. And their goal was to insert this entire massive library of variants into 11.4 million individual cells.
Wow, 11.4 million.
Right. So they could observe them all simultaneously in a single pool. And to get the genetic material inside the cells, they used this molecular tool called piggyback transposition.
Piggyback. Okay, I'm trying to picture this. Is piggyback functioning like a molecular Trojan horse, like something sneaking the new DNA sequence past the cell's defenses?
Trojan horse is close, but it's less about trickery and more about, like, a highly regulated payload delivery. Piggyback is what we call a transposon system.
Like a jumping gene.
Exactly, a jumping gene. It acts as this specialized delivery service that efficiently carries the mutant gene sequence and integrates it directly into the host cell's genome.
Okay, so it drops the package right into the DNA.
Right, but the crucial part of this step is maintaining a really low multiplicity of infection.
Which means what exactly?
It means they carefully control the concentration of these delivery vehicles. They have to make sure that each cell only receives one single variant to express.
So you don't want one cell getting like 5 different mutations because then you would know which one caused the problem.
Exactly. It would ruin the data. And they performed this delivery across diverse cell models. They used human bone cancer cells, induced pluripotent stem cells, and even stem cells they coaxed into becoming neurons.
Okay, so now we have this giant pool of 11.4 million cells, and each one is expressing just one of the 3,000 different mutations.
Yep.
But they are all mixed together in this massive microscopic soup. How on earth do you know which specific cell contains which specific mutation when you're staring through the microscope?
Yeah, that was one of the major hurdles they had to solve. So typically researchers attach a piece of RNA to the mutant gene to act as a linear barcode tag.
Like a molecular name tag.
Right. The idea is that you can just read the RNA to identify the cell. However, cells have evolved these powerful defense mechanisms against free-floating linear RNA.
Because that's what viruses look like, right?
Exactly. A lot of viral material is linear RNA. So the cell's exonucleases, these little enzymes, they recognize the exposed ends of the linear barcode and they just chew it up.
Like a microscopic paper shredder.
Exactly like that. In fact, in these experiments, using linear barcodes resulted in 0 or maybe just 1 sequencing read in 56% of the cells.
So more than half the time, the name tags were degrading before they could even be read.
Right. The cell thinks it's fighting off an infection and destroys your tracking device.
That makes total sense. So what was the workaround?
It's so clever. They engineered the RNA barcode to fold into a continuous circle.
A circle, so there are no ends.
Exactly. Because it has no exposed ends, the cellular exonucleases have nothing to latch onto. It makes the barcode highly stable.
Oh, that's brilliant.
But the real genius comes when they amplify it. They use an enzyme for a process called rolling circle amplification.
Rolling circle amplification. How does that work?
Well, because the barcode is a closed loop, the enzyme just goes around and around the track, endlessly copying it over and over. It creates this massive repeating chain of the exact same barcode sequence.
Oh, wow. So it's like instead of handing out a single paper flyer that easily gets lost or shredded, they painted the barcode on this massive, continuously looping billboard inside the cell.
Yes. So there is absolutely no mistaking its identity. This single innovation gave them a 13-fold increase in sequencing reads inside the cell.
The 13-fold increase. That's wild. Okay, so once every cell has this indestructible circular name tag, we get to the visual part, right?
Exactly. The visual profiling relies on a technique known as cell painting.
Cell painting? That sounds like art class.
It kind of looks like it under the microscope. Researchers use a cocktail of fluorescent dyes to stain the different architectural components of the cell, making them glow.
So they're lighting up the cell's anatomy.
Right. They use DAPI, which is a blue fluorescent stain, to tightly bind to the DNA and illuminate the nucleus. Then they use phalloidin to light up the actin microfilaments.
And actin, those are like the structural tension cables, right? The things that give the cell its shape.
Exactly, the tension cables. Then they use wheat germ agglutinin to highlight the cell's outer plasma membrane and the Golgi apparatus.
Okay.
And finally, a specific mitoprobe to illuminate all the mitochondria.
So you have this highly detailed, brilliantly glowing map of the cell's entire physical structure.
Yes.
But, I mean, visually inspecting 11.4 million of these glowing cells.
Yeah.
That's impossible for a human to do.
Yeah.
How does that data actually get crunched?
Oh, they rely on some heavy computational lifting. They developed this custom software called StarCall.
StarCall.
Right. And its job is to align all those millions of barcode sequencing reads with the physical photographs of the cells.
So matching the billboard name tag to the mugshot.
Precisely. And once the mutation is matched to the specific cell image, another open source software suite called Cell Profiler takes over.
And what does Cell Profiler do?
It doesn't just look at the image, it extracts over 1,000 highly specific morphological features.
1,000 features from one cell.
From every single cell. It calculates the nuclear area, the exact granularity of the mitochondria, the texture of those actin cables we mentioned, the overall symmetry of the cell.
That's an insane amount of data.
It really is. It generates a high-dimensional mathematical map, what they call a morphological profile, for every single one of the 3,000 variants.
Okay, before we see what happens when they unleash this on an actual gene, I just want to pause. Let's do a quick 5-minute recap for you listening, just to make sure our mental model is perfectly clear.
Good idea. There's a lot of tech here.
Right. So bullet point 1, we moved away from simple 1D tests that just ask if a cell lived or died, and instead we're tracking high-dimensional physical features.
Exactly.
Bullet point 2, to pull this off, we use a payload system to drop single mutations into millions of cells, and we tag them with looping RNA billboards, the circular RNA barcodes, so they don't get destroyed.
Right, solving the identification problem.
And bullet point 3, we use fluorescent dyes in this cell painting process, plus machine learning, to map out 1,000 structural data points per cell, giving us an exact morphological profile.
That is a perfect summary. We essentially built a massive spatial diagnostic machine.
So to the listener, think about your own body for a second. If a single changed letter in your DNA changes the literal physical shape of your cells, how many different ways could that go wrong?
Oh, so many ways.
Right. Let's look at the first example. Let's get into LMNA. If we point this high-dimensional camera at the LMNA gene, what are we actually taking a picture of?
So the LMNA gene produces a protein called lamin A. And you can think of lamin A as the critical steel meshwork-like scaffolding that lines the inner membrane of the cell's nucleus.
Okay, so it's the support beams for the nucleus.
Exactly. It dictates the overall shape of the nucleus and provides the mechanical stability needed to protect all the precious DNA inside.
That sounds pretty important.
It is. When mutations occur in this gene, they cause a group of severe, seemingly unconnected diseases collectively known as laminopathies.
Unconnected how? Like different symptoms?
Very different. Depending entirely on the specific mutation, a patient might develop muscular dystrophy or severe forms of cardiomyopathy.
Heart disease.
Right. Or even progeria, which is a really tragic disease that causes rapid premature aging in young children.
That is always the thing that completely blows my mind about genetics. One single gene acts as the instruction manual for one protein. But depending on which paragraph has the typo, you either get a heart condition or you literally age a decade in a single year.
It's astounding.
So when they ran the 1,700 different LMNA variants through the VIS-seq protocol, what did the cell profiler software actually find in those pictures?
Well, out of the 1,000 different structural features they tracked, the software identified a set of what they called hit features.
Hit features.
Yeah, the specific measurements that were most consistently perturbed or messed up by the mutations. For lamin A, the dominant features were the intensity of the protein, its tendency to form useless clumps or aggregates, and nuclear circularity.
Nuclear circularity, meaning like how perfectly round the nucleus was.
Exactly, how perfectly round it was.
Now, normally when we talk about mutations, we assume they act as a loss of function, right? Like the mutation breaks the part, and I'm assuming some of the variants did just that. The scaffolding collapsed and the nucleus became crumpled and deformed.
Yes, many did exactly that.
But I know VIS-seq revealed something incredibly counterintuitive about that nuclear circularity feature.
It really did. While many variants caused exactly the loss of function you described, crumpling the nucleus VIS-seq identified a highly distinct cluster of variants that did the exact opposite.
The opposite.
They actually increased nuclear circularity. They made the nucleus exceptionally perfectly round.
Yeah.
In genetic terms, this is a gain of function. The protein isn't broken in a way that stops it from building a structure. It's broken in a way that builds a completely altered, hyper-round structure.
See, this is where my intuition just hits a wall. If lamin A is the steel scaffolding meant to keep the nucleus in shape, wouldn't a perfectly round, hypercircular nucleus be a good thing?
You would think so.
Right. Why does making a cellular structure too perfect result in a devastating disease?
It all comes down to the physics of the cell. To understand why a rigid circle is pathogenic, we have to look at the 3D architecture of the lamin A protein itself.
Okay, let's zoom in on the protein.
Lamin A is composed of a long central rod domain, which is made of these rigid coiled coils. Specifically, they're called coil 1B and coil 2A.
Okay, rigid coils.
Right. And connecting these rigid coils is a short, highly flexible hinge region known as linker 12.
So the protein itself is structured a bit like a medieval flail or maybe nunchucks. You have these rigid rods connected by a flexible chain segment.
That visual works perfectly. So VIS-seq mapped the physical shape of the cells directly back to these specific protein domains.
And what did that show?
It revealed that if a mutation occurs in the rigid rods, the coiled domains, the proteins fail to assemble properly. The structural integrity just fails, they form those useless aggregates, and the nucleus crumples.
Okay, so a broken rod equals a crumpled cell.
Exactly. But if the mutation hits that flexible hinge, the linker 12 domain, the proteins still manage to assemble into scaffolding. However, they lose their mechanical flexibility.
I always see.
The resulting meshwork forces the nucleus into this hypercircular, hyper-rigid shape.
And a rigid shape can't absorb stress.
Precisely the issue. Biological systems require dynamic flexibility.
Yeah.
Think about a cell in a beating human heart.
Right.
Every single time the heart muscle contracts, the cells undergo immense mechanical shear stress. The nucleus must be able to stretch, compress, and rebound.
Like a shock absorber.
Exactly. If a mutation in the linker 12 domain makes the nuclear envelope hypercircular and brittle, it can't deform under pressure. The mechanical stress literally causes the nuclear envelope to rupture.
Oh, wow.
Which damages the DNA inside and directly leads to the death of the heart tissue.
That is just a stunning level of insight. I mean, we aren't just saying the gene is bad. We are proving that breaking the rigid structural rod causes a crumpled nucleus, while breaking the flexible hinge causes a brittle, hyper-round nucleus that literally shatters under the stress of a beating heart.
It's incredible resolution.
That explains perfectly why different mutations in the exact same gene lead to vastly different physical outcomes for a patient.
And that is exactly the power of moving beyond a simple binary classification. VIS-seq grouped these variants into 10 distinct clusters based on these physical effects. Disruption of different subdomains leads to wildly different cellular fates.
Okay, so that's LMNA. Let's do another quick recap for you, the listener, before we move on.
Sounds good.
Bullet point 1. LMNA controls the cell's nuclear scaffolding. Bullet point 2. VIS-seq grouped variants into distinct clusters based on their exact physical effects. And bullet point 3. Mutations don't just break the nucleus. Some make it hypercircular, mapping directly to specific 3D protein subdomains, like the flexible linkers versus the rigid coils.
Spot on. But, the LMNA gene is primarily a structural story.
Right, scaffolding.
The implications become even more profound when we apply this high-dimensional imaging to genes that control cellular communication and decision-making.
Which brings us to the second gene the researchers analyzed, PTENN. So, as a question for you listening, if changing a protein's physical shape causes heart disease, what happens when a mutation alters a protein that acts as an intracellular messenger?
It gets really complicated really fast.
So, if LMNA is the physical steel scaffolding of the cell, what is the function of PTEN?
PTEN is a phosphatase. It's an enzyme that acts as a master regulator. Its primary role in the cell is to suppress a massive signaling cascade known as the PI3K-AKT-mTOR pathway.
Okay, PI3K-AKT-mTOR. That is a mouthful. What does that pathway actually do?
You can think of that pathway as effectively the central command for cellular growth, proliferation, and survival.
So if that pathway is the engine driving the cell to constantly multiply and consume resources, PTEN is like the heavy foot on the brake pedal.
Exactly. It keeps that growth in check so things don't spin out of control. Without PTEN functioning properly, the cell receives a constant, unchecked signal to just grow and divide.
Which sounds a lot like cancer.
It is. This mechanism explains the clinical history of PTEN mutations. Inherited mutations in this gene frequently cause a group of conditions known as PTEN hamartoma tumor syndromes, or PHTS.
Right, which makes logical sense. You remove the brakes, cells grow endlessly, and they form tumors.
Yes, but there is a profound clinical mystery here. PTEN mutations are also a heavily documented cause of autism spectrum disorder.
Wait, autism?
Yes. Frequently presenting with macrocephaly, which is an enlarged brain size and developmental delay.
And that is the paradox. How can one single gene, responsible for suppressing growth, cause severe tumor syndromes in one patient, but manifest as a neurodevelopmental disorder like autism in another patient?
For a long time, the prevailing theory in the medical community was that it was simply a matter of severity.
Like a sliding scale.
Yeah. The assumption was that the variants causing autism were just hypomorphic. In other words, they were just milder, weaker versions of the mutations that cause tumors.
Okay, I actually heard this described like a leaky faucet versus a totally burst pipe.
Yes, that analogy was used a lot.
Like the autism variants let a little bit of unregulated growth slip through, causing maybe brain overgrowth during development, while the tumor variants were a total structural failure of the pipe.
Exactly. The brake pedal was still somewhat functional, just not as strong as it should be.
But the VIS-seq data completely dismantles that leaky faucet theory, doesn't it?
It does. It proved that analogy is fundamentally flawed. To understand how, the researchers engineered over 1,200 different PTEN variants into neural stem cells.
Okay, 1,200 variants.
And they trained Cell Profiler to track 3 primary features, which we can think of as the protein's abundance, its activity, and its localization.
Abundance, activity, and localization.
Right. They measured how much of the protein was present, how effectively it was stepping on the brakes, which they measured by a downstream signaling molecule, and exactly where the protein physically resided inside the cell.
Okay, so when they mapped those 3 things across the tumor variants and the autism variants, what was the big differentiator?
The breakthrough was entirely in the localization.
The localization.
Where it was, right. While the autism-associated variants did generally show, milder impairments in overall abundance and brake activity compared to the severe tumor variants, that wasn't the defining characteristic.
Okay.
VIS-seq revealed that the ASD-associated variants were physically mislocalizing. Instead of staying in the cytoplasm where they normally regulate that growth pathway, the PTEN proteins were aberrantly migrating into the nucleus of the cell.
Wait, let me make sure I'm getting this right. So it's not just a weaker brake pedal. The tumor variants break the brakes entirely, but the autism variants unbolt the brake pedal and, move it into the trunk of the car where it doesn't belong.
That's exactly what's happening.
But why does that specific movement cause a neurodevelopmental condition?
This is where the spatial context is just vital. In the cytoplasm, PTEN's job is halting growth. But if you inappropriately traffic that protein into the nucleus, it finds itself in a completely different molecular environment.
And the nucleus houses the DNA, the genome.
While research is still ongoing, we know that nuclear PT interacts with completely different pathways. It's implicated in maintaining genomic stability, regulating chromatin structure, and potentially altering the expression of other genes.
Oh, wow.
So during fetal brain development, a mislocalized PTEN isn't just failing to stop growth, it is actively interfering with the complex genetic choreography required to wire neural circuits properly.
That is absolutely mind-blowing. The physical location of the protein alters its entire functional identity. How distinctly could the researchers see this mislocalization in the images?
It was incredibly clear. They measured it using a specific feature called DPI-PTEN correlation.
DPI. That was the blue dye for the nucleus we talked about earlier.
Exactly. If the glowing signal of the PTEN protein strongly overlapped with the glowing signal of the DPI stain, it meant the protein was trapped inside the nucleus.
Ah, so they literally just looked for where the glowing colors mixed.
Exactly. And this single physical feature was so robust and so distinct that the researchers were able to train a machine learning classifier on the cell images.
And I want to highlight the numbers on this classifier because they are staggering.
They really are.
The researchers trained this algorithm to distinguish between a tumor-causing variant and an autism-causing variant based solely on the structural photographs generated by VIS-seq, and the algorithm achieved an accuracy rate of 92%.
An area under the curve of 0.92.
It could literally look at the shape and location of the mutation and predict the clinical disease with 92% accuracy.
It's phenomenal. And to truly appreciate how impressive that 92% is, we have to look at the current alternative. Right now, clinical geneticists use state-of-the-art computational predictors, programs like AlphaMasense, Revel, or EV.
Right, to guess the impact of a variant.
Exactly. These AI models are trained on evolutionary conservation and 1D amino acid sequences. They essentially look at a typo in the code and guess if it's dangerous based on how rare it is in evolutionary history.
Okay, so purely text-based prediction.
Right. But when they applied these standard predictors to the variants in this study, they performed significantly worse. In fact, for the structural LMNA gene we talked about earlier, the computational predictions only correlated with the actual physical cellular reality at a rate of 0.27 to 0.35.
Wow, that is terrible. But it makes sense because an algorithm that only reads text can't predict a 3D structural collapse.
Exactly.
It might know a typo is bad, but it has no way of knowing if the typo makes the protein clump together into a useless ball, or if it makes a scaffolding violently hyperrigid, like with the linker 12 mutations.
Exactly. A 1D sequence model assumes any change in a vital region is equally destructive. It ignores the specific physical flavor of the destruction.
Let's do our final 5-minute recap here.
Good call.
So, bullet point 1, PTEN mutations cause both cancer and autism, which has historically confused doctors. Bullet point 2, VIS-seq showed these aren't just points on a 1D scale of badness. And bullet point 3, autism variants cause the protein to get lost and mislocalize to the nucleus, which is a completely distinct mechanism from the tumor variants.
Perfect.
So for you listening, why should we care? Think about how this changes personalized medicine. If your doctor knows exactly how your specific mutation misbehaves, physically, how much more targeted could your treatment be?
It's the future of medicine. What this paper definitively proves is that we cannot accurately model human biology using only sequence data. We desperately need multimodal, high-dimensional ground truth data.
Like the morphological profiles generated by VIS-seq.
Exactly. This spatial data is what must be used to train the next generation of predictive AI models, bridging that gap between a genetic sequence and a physical disease.
It completely shifts how we have to think about genetics moving forward. Genetic variants don't exist on a binary scale of benign or pathogenic.
No, they don't.
They live in a rich, multidimensional continuum. Every mutation has a shape, a physical location, and a dynamic mechanical reality.
And by integrating technologies like pooled cell tagging, fluorescent imaging, and machine learning, we are finally developing the vocabulary necessary to describe that multidimensional reality. We are moving from a static code to a dynamic spatial architecture.
It's just incredible. VIS-seq literally paints a high-definition portrait of mutation's consequence.
It really does.
So as we wrap up today's deep dive, I want to leave you with a final thought to mull over. There are over 5,400 known disease-associated human genes out there.
That's a lot of genes.
It is. If VIS-seq can definitively untangle the difference between a cancer-causing variant and an autism-causing variant in just one gene, simply by tracking where a protein wanders off to, what other long-standing medical mysteries are out there?
It's a huge question.
How many conditions do we currently lump together under one umbrella name, treating them with the exact same blunt therapies simply because we haven't taken a picture yet? How many diseases are actually a dozen distinct mechanical failures, just waiting for us to turn on the light and look? Thank you for joining us on this deep dive into the source material. Keep questioning the physical reality of the world around you. Keep exploring those gray areas, and we will see you next time.